Hello guys, welcome to our channel. Today we are going to discuss as a part of the mission 200 plus, we are going to discuss today the physiology subject. So guys, we are going to cover almost all the important topics for the PYTs or the essential topics. Please stay tuned. And those who are interested to buy these notes, this is a paid content. And if you are interested, please text to this Telegram ID. And from you can join the Telegram main group, and also from there also you can text me, so that we can add you into the other group that is only containing of the annotated and the unannotated copies of this lecture. So let's start the lecture, guys. Those who are new to this channel, please do subscribe and share with your friends. So let's start with the first discussion. So can anyone please identify these individuals and comment in the comment box. So the first thing that I want you to know, guys, is this is the father of the physiology. So he is known as Claude Bernard, right? So he is the first scientist who is modern physiology. He is the father of modern physiology, known for his many important media, interior, and also how the internal system and the other physiological structures will be working together. He have explained so many years in the very late 18th century only. He have explained. So he is a Claude Bernard, and I hope you guys all might have already heard about the Guyton textbook, right? So the Guyton famous textbook Physiology is the most common copy that has been used by the international and in our India also. So this person is known as a Guyton. So such an inspirational story. Actually, he wanted to become the cardiothoracic surgeon, but unfortunately, when he want to take the admission in the Harvard High School, he has suffered from the polio paralysis. You can see he was suffered from the polio paralysis. After that, he has to discontinue because once the polio person paralysis, his upper arms also severely affected. So he cannot perform any surgery. So he then shifted to the physiology, and he didn't give up his dream, and after the many research he have published the Guyton, and today still the Guyton is one of the most appreciated and most sold textbook in the medical fraternity. So we are very thankful for the Guyton, and also take the inspiration from the Guyton and the Claude Bernard, because they are the most rejected persons. They have faced many failures, but still they contributed what they can do and what they can help for their future and also for our future, right? So we don't know whatever chance we get, whatever the personality we are, but what we all can prove, and we all can create our own history. Okay, so please don't give up. There are many plenty of the chances, and everything will come at once, and we will have that beautiful life. Okay, don't worry, guys. So let's start the discussion, guys. So first and foremost important, we are going to discuss about the pause feedback system. So we have the positive feedback system, and we have the negative feedback system. What is mean by the positive feedback system? Positive feedback system means it will push further. Okay, it will push. Whereas the negative feedback mechanism, it will push, but more of it will pull back. Okay, so how example, let's see. So the most of the example that we see is the, what is this TRH? From the hypothalamus it will give the TSH, and then it will give to the T3 and T4, right? So this is the positive feedback mechanism to sending to make it. Once this is reached the normal levels, what does this T3 T4 will do? It will go back to the TSH and tell, please stop, that the already production is enough. So this is what the negative feedback mechanism. Okay, so it is telling that already our physiological role is done, we have to stop. If not, it going to exceed and can trouble the person. So this is what the negative feedback mechanism. What about the positive feedback mechanism? For example, you have the injury. Okay, once the injury is done, what happens? There will be the clot formation, and after the clot formation, the bleeding is going to stop, right? So even if you remove this, whatever the clot is already formed, even if you remove the clot, again the clot will form, right? So this is what the positive feedback mechanism, it is pushing further to make the normal. So some of the examples for the here, this is the LH search, action potential, clotting, and also parturition. These are the some of the examples. Next, for the negative feedback mechanism, mostly all the hormones like insulin, glucagon, thyroid, and also we have the baroreceptors. We will be discussing in the cardiac topic, guys. So baroreceptors, these are the some of the examples. And next we also have the other one, that is the feed forward. Okay, feed forward. So what is mean by the feed forward? This is a type of anticipatory. What is mean by the anticipatory? For example, if you see or if you heard about the food ingredients, food types, immediately you have the saliva production, right? So this is one of the anticipatory. And some people, whenever they wanted to do the exercise, there will be increased heart rate. So these kind of the things are the anticipatory, and this is the feed forward mechanism. All right, next moving on to the cell and organelles. This is the most important, and directly we can see the very simple points that we are going to discuss now. So this is the endoplasmic reticulum, and here you can see this is the nucleus, mitochondria, Golgi complex, cytoplasm, lysosomes, cytoskeleton. Let's see one by one. So first we will be discussing about the nucleus. So what is the important of the nucleus, guys? It will be contains the main DNA structure, and this DNA storage is person to person, it is going to be differ. So this is the what the most of the importance of the cell is. The nucleus will maintain the DNA storage. Next, plasma membrane. So plasma membrane contains of the three important structures. One is the proteins, and other one is the lipids, and then carbohydrates. So in this, the most common will be the lipids, then this is the second most common, and this is the third most common, which will be presenting in the plasma membrane. Next, endoplasmic reticulum. We will be seeing the endoplasmic reticulum. One is the smooth endoplasmic reticulum, other one is the rough endoplasmic reticulum. So rough means it will be contents of the ribosomes. Okay, so these ribosomes which will be helping for the protein synthesis. This will be helping for the protein synthesis. Not just the protein synthesis, it will be also helping the protein in the folding. So while folding, what is the thing that we are needed? That is the chaperones. So chaperones is needed for the protein folding, and also protein degradation. Okay, protein degradation is also seen here with the help of the ERAD. So this is the rough endoplasmic reticulum. Smooth endoplasmic reticulum, S for smooth, S for steroid synthesis. Okay, it will help in the steroid synthesis. It will be helping the calcium storage. It will be also help in the drug detoxification. In the liver, it will be helping for the drug detoxification. Next, we have the Golgi apparatus. So if you observe the Golgi apparatus here, so this is the cis end, this is the trans end. So what is meant by the cis and trans? So once the Golgi apparatus, any system, any protein or any process is happening, they enter via the cis and they will make the post-translational modification. They will have post-translational modification. And after this post-translation modification, from the trans, they are going to go out. It's like packing. Here it is going to be the packing and making and then sending its important material. Okay, this is the Golgi apparatus. Next, mitochondria. Mitochondria, which will be contents of the, again it will be contents of the double stranded DNA. Okay, and almost 16,500 base pairs it will be containing, and most of the mitochondria is important for the ATP production, right? This is important for the ATP synthesis, and it is only transmitted from the mother to children. This is the only pattern coming from the mother to children. It is not coming from the father or any other structure. It is only coming from the mother. Next we will be also discussing about the lysosome. Lysosome is known as a suicidal bag, right? This is known as a suicidal bag because it is going to degrade or remove the any other structures that is available. For example, it can go into the autophagy. What is mean by the autophagy? If you are in the starvation, it will go eat by itself and it will release the energy from the after eating those mitochondria structures. This is the autophagy. And what is the enzyme that it will use? It will use the acid phosphatase. Okay, acid hydrolase. These are the enzyme which is going to use. Next, we also have the peroxisomes, right? Peroxisomes are important for the oxygen radicals. So to kill the oxygen radicals with the help of the catalase enzymes, it is going to helpful. Next is also going to important for the very long chain fatty acids oxidation. So if there is a defect in the very long chain fatty acid oxidation, so the disease is most commonly presented with a Zellweger disease. Recently asked in the NEET PG, INICT and the Fmge also. This is the Zellweger disease. Okay, so these are the important points that you have to know, and the most importantly, some of the markers that you have to know. So the markers such as plasma membrane, which will be contents of the sodium potassium pump. This is the plasma membrane form. And for the endoplasmic reticulum, this is the G6P, glucose 6 phosphate. And for the Golgi apparatus, you will be having the galactosyl transferase. Okay, and mitochondria, it is ATP synthase. For the mitochondria, it is ATP synthase. And here, acid phosphatase for the lysosome and catalase for the peroxisomes. All right, next, some of the important points that I have to discuss here is about the plasma membrane, sorry, cytoskeleton. So cytoskeleton, which will be having the three types. Okay, so first it will be having the microfilaments. Okay, microfilaments. It will be having the filaments, intermediate filaments, and the most importantly, it will be also consisting of the very important structures such as the cytoskeleton, which will be containing of the microtubules. Okay, so these are the important structures that will be present in the cytoskeleton. So let's see one by one. Microfilaments, this will be microfilaments which will be, so this is first one, second and third. So first one will be contents of the myosin and the actin. So why these both are important, guys? These both are important for the mostly for our own body structures like muscles, muscle contraction, it is important. So intermediate filaments, we have the vimentin and we have the keratin. So and also some other such as the desmin. These are important for the tumor markers we are using today. And next, the third one is the microtubules. In that we have the dynein which will be helping for the anterograde transport, and for the kinesin it will be for the retrograde. And tubulin, tubulin is going to have the mitosis, continuous mitosis will be seen. So if you block this tubulin, we can use this as a drugs, right? One of the example is vincristine. Okay, these are the some of the example. So I want you guys to just take note. Dynein is a retrograde and kinesin is the anterograde. Okay, so these are the some of the important points that you have to take note, especially for the cell membrane. Okay, next, here the cell junctions. So in the cell junctions, just take note, they are important cell junctions, and even the INICT also asked recently about the gap junctions. So where are the gap junctions are present and what is importance of the gap junctions? So let's go by the one by one. First we are going to discuss about the tight junctions. So tight junction location has been asked, that is the apical lateral. Okay, that is the tight junctions. And what does it contains? It contains the claudin and also occludin. And where this is presented? This is presented in the blood brain barrier, blood spermatic barrier, renal tubules, and also corneal endothelia. Very tight structures it is going to present is the tight junctions. Desmosomes will be present in the rough areas, and what it is going to contains? Desmoglein. It is going to contains the desmoglein. Examples: rough structure areas such as the skin, and also more prone to injury, and that is the cervix. Okay, and adhering junctions. In the adhering junctions, it will contains the catherines. It will be contains the catherines, and these catherine junctions is also present in the mostly in the skin and other structures. Examples: it is present in the GI also. Okay, GIT. Next, gap junctions. Gap junctions is contains of the connexon, contents of the connexon. An example: it is present in the GIT, and also it is present in the heart. Especially heart, it is very important. In the heart, it is going to form the one of the most important structure, that is the functional syncytia. Okay, functional syncytium is formed by the gap junctions. Okay, these are the important points that you have to take note about the junctions. Next, here are the types of the cell signal. Autocrine, it is for the self cell only, and for the paracrine it will be contents of the nearby cell, and for the signaling for the across the gap junctions, here you can see this is one of the example for the heart where the signaling will be gap junctions only, and endocrine with the help of the bloodstream. Next, coming to the secretion of the different exocrine glands. For the merocrine, you are going to have the secretory vesicle. For the apocrine, it will be pinched off, or the portion of the cell that will be pinched off. Next, the holocrine, it will be maturely, it is going to produce the different types of the secretory products. Okay, this question has been also asked in the examination. Next, moving on to the secondary intracellular receptors. Okay, so how the receptors is going to work. So body contains of the many receptors. In that we have to take note about the cytoplasmic receptors. What are the cytoplasmic receptor, guys? The main example for the cytoplasmic receptor is the cholesterol, and we also have the vitamin D and steroids. These are the some of the examples for the cytoplasmic receptors. And next we also have the nuclear receptor. Nuclear receptors, vitamin A, thyroid is main examples. Other than that, we also have the estrogen and progesterone. Okay, this has to be taken. Next, moving on to the secondary signaling. Okay, secondary messengers. So in the secondary messengers, we have the examples such as ion mediated, such as NMDA. This is one of the example. And for the camp, we have the all the hormones such as TSH. We have ACTH, okay, ADH, LH. These are the camp. Okay, with the help of the adenine cyclase. CGMP. This is for the nitric oxide, natriuretic peptides. Okay, and also BNP, brain natriuretic peptides, and calcium related. In the calcium related, we have the colmodulin and TRH. Okay, TSH is in the CAM and TRH is in the calcium. Please take note that. Okay, and next we also have the some of the pathways that you have to take note. Tyrosine pathway. Tyrosine pathway is for the insulin, and we have the JAK-STAT pathway. Okay, JAK-STAT pathway is for the growth hormone, also for the prolactin, leptin, and most importantly erythropoietin. Okay, JAK-STAT, this question has been also asked in the examination. So these are the secondary messengers and important structures, receptors also that we have discussed. Next, moving on to the total body water, how much that the body is going to contain. So in the total body water, we have almost 60% of the body content will be water only, and it is subdivided. 2/3 will be the ICF and 1/3 will be the ECF, extracellular fluid. And in the extracellular fluid, again the 2/3 will be interstitial, 1/3 will be the plasma total that we are going to contain. And we have to be calculating this. How we are going to calculate, with the what help? So total body water, which will be calculated with the help of the deuterium, or we can use also tritium. Okay, next we have the extracellular fluid, which can be calculated also. We can use the different types such as inulin and also sucrose, mannitol we can use for the extracellular fluid. And we can also calculate the plasma with the help of the Evans blue. Okay, Evans blue and also albumin. Whichever the plasma contains, that structures we can use. Albumin also we can use. How we can calculate the ICF? ICF, we cannot calculate the ICF. What we can do? We can minus this both, right? Total body water minus ECF. We can get the normal value. And we cannot calculate the interstitial fluid. So for the interstitial fluid, ECF minus plasma, right? This is the best way we can get these structures calculated. Next, we have the sensible loss also. So sensible loss, which will be from the sweat. Okay, from the sweat. So around the 100 ml per day we can say from this one. And non-sensible also we have. Okay, non-sensible, which will be going out of the lungs, especially and skin. So this is around 500 ml per day. Okay, this is the sensible versus a non-sensible type of the presentation that we are going to see. Next, here the another image that you can see, this is the Dravet-Yannet diagram. So in the Dravet-Yannet diagram, we are going to see how the presentation will be presented, how the volume contraction or the volume expansion seen if there is a presentation with a disease and after the fluid intake. So in the isotonic, there will be the same, right? So what is the normal osmolarity first? So the normal osmolarity will be around the 295 to 300 milliosmoles, right? How we are going to calculate? 2 sodium plus glucose by 18 plus urea 2.8. So this is how we are going to calculate the osmolarity. So when we add the osmotic isotonic fluid, it will be the same content only, right? So the content of the volume expansion will be the same for the isotonic fluid, and whereas the hypertonic fluid, so the ECF will be increased and same as the ICF will be increased in the volume expansion, and the hypotonic also, so there will be increase in the expansion. Here you can see this is the expansion of the fluid, expansion will be seen, and in the hypertonic versus the hypotonic, the difference will be here the osmolarity will be also increased, and here the osmolarity will be decreased. So this is the difference between the hypertonic and the hypotonic. What is happening? Just by that only you can answer these questions. For example, I given the isotonic. What is the most commonly isotonic that we are going to use? 0.9% NS, right? This is the isotonic. So the cell contains the normal osmolarity 295, and whatever the fluid that we are giving also same contents. So there is no change. So only the easy permeability will be the water only. Next, whereas the hypotonic fluid. So hypotonic fluid, when I am giving this 0.45% of the NS. So this 0.45% of the NS, which will be contains the cell, inside the cell will be contents of the more. Whereas the fluid that I am giving is contents of the less solution. So what happens? Most of the cell will be shifting, the water will be shifting here. So end of the day, what happens? This cell will be swell. Okay, this cell will be swelling here. Whereas the hypertonic, so for example D5, so when we are giving, the cell contains itself very less, and we the solutes that we are giving almost full of the solutes. What happens? The end of the day, the shell will shrink, the cell will be shrinking. So these are the different changes that we are going to see in the, so these particularly the fluids and their electrolytes. Okay, next I also want to guys to understand about the carrier proteins. What are the carrier proteins that will be present? This is the matching that has been asked in the examination. Carrier proteins. What are the carrier proteins? We have the thyroxine. For the thyroxine, pre-albumin is most commonly going to carry them, and free fatty acid. So for this albumin, and for the hemoglobin, we have the haptoglobulin, and for the heme, it is the hemipexin. These are the carrier proteins that will be carrying different types of the important structures in our body. Okay, next moving on to the transports. Primary active transport versus the secondary active transport. So basically when I say active transport, it is going to use the ATP. So that is the clear, right? So it is going to use the ATP. So the most common example which will be we are discussing in the primary will be the sodium potassium pump, right? Sodium potassium pump. So how many sodium is going out? So the 3 sodium will be out. How many? 3 sodium will be out. How many potassium will be in? The 2 potassium will be inside, along with the ATP production that will be seen here. So this is what the most importantly sodium potassium pump, and how this is presented? Antiport, the two different directions, that is known as a antiport. Next, secondary active transport which will be coming from the symport. What is the example? SGLT, right? SGLT, sodium glucose transporter. So whereas the sodium and the glucose facilitated together to transform or come inside the body, that is the SGLT receptors. And what is the common mechanism that is seen with the ORS? Whatever the ORS we are giving for this children, that is actually carried into our body with the help of the sodium glucose transporter only. Okay, that is the ORS mechanism. This is a secondary type. And next, coming to the passive type. Passive type means there is no need of the ATP. There is no need of the ATP that we have to work on. So what else we have the examples? One is the simple diffusion. So the example will be the Fick's law, right? Where there will be the only oxygen plus the uh, gaseous exchange, which will be seen here in the simple diffusion, because there is no need of the any carrier protein. Next, facilitated diffusion. In the facilitated diffusion, you need the carrier. So you need the carrier. Examples such as glut, okay, different types of the gluts, and also aquaporins, and also most of the amino acids. They need the carrier protein to facilitate. Still they don't need the ATP, but they need the carrier protein. So this is the osmosis. This is a semi permeable, just now we discussed. So these semi permeable, which will be only carrying the ions, water, that will be carrying forward from the osmosis, right? These are the some of the examples that you have to take note, right? Next, another point that I want to take note, you guys, these are the important topics that has been asking in the examination. Don't ignore that this is just a small chapter, but this small chapter can include the many types of the questions. Another graph that I wanted to take note, you guys, if there is a simple diffusion, there is no saturation kinetics. But if there is any other saturated kinetics, for example, this is the facilitated diffusion, this is the simple diffusion, so the saturation will be seen, right? This is one of the graph is also asked in the examination. Next, moving on to the neurophysiology. Here, moving on to the neurophysiology that we are going to discuss now. In the neurophysiology, you have to take note about the cells first. So this is the ependymal cells, and what is this ependymal cells which will be doing? This is important for the CSF production. This is important for the CSF production. Microglia, this is the macrophage of the brain. This is the macrophage of the brain. And next is squam cells. Squam cells will be contents of the myelin sheath. Okay, squam cells are important for the myelin sheath, and the more the myelin sheath, more the nerve conduction, right? So the more nerve conduction will be seen, and the most of the nerves will be present in the brain or the peripheral system. In the peripheral system, so the squam cells is present in the peripheral system. And satellite cells, the name itself tells satellite and supporting. S for satellite, S for supporting cells. Next, astrocyte. Astrocytes has a many major roles. Astrocytes is like a nourishment. These are the important it will help in the nourishment and the diseases that, and also it will maintain the neurotransmitters. Those who are new to this channel, please do subscribe or not, and also help in the CSF maintenance. CSF maintenance also astrocyte will be maintained. And oligodendrocytes. This is also contents of the myelin sheath, and this is very importantly present in the brain. Okay, CNS. It will be contents in the CNS, the structure that will be consisting of the present in the brain and the CNS. Okay, this is the important structures that you have to take. Next, coming back to here, what is the upper motor neuron and the lower motor neuron? From the brain to the spinal cord, that is the upper motor neuron, and from the spinal cord to the targeted organ, that is the lower motor neuron. And if they are the lesions, if they are lesions, for example, upper motor neuron, what is the presentation, and the lower motor neuron, what is the presentation? In the upper motor neuron, actually the upper motor neuron is has to decrease the tone. But if the upper motor neuron is already have the lesions, what happens? The tone will be increased, right? The main function only you have to decrease the tone, but if you are not at all working, what happens? The tone will be increasing. The lower motor neuron, the tone will be decreased. And here the deep tendon reflexes will be exaggerated. Here the deep tendon reflexes will be decreased. And Babinski sign will be the positive, like dorsiflexion of the big toe, that will be seen here. And here the Babinski sign is negative. And what happening here? Upper motor neuron, only tone. The tone is increasing, increasing, increasing. So there is no chance of the rest at all. So atrophy will be not seen in this patient. Whereas here, tone is decreased. There is no movement at all. So obviously the patient will be having the atrophy. Okay, this is the major difference. What is the example here, guys? Spastic paralysis, and here flaccid paralysis. Complete tone is decreased. These are the some of the upper motor neuron and the lower motor neuron lesions. Okay, next. Next, I want to discuss with you guys about the receptors, skin receptors. Just take note of the Merkel disc, Messner corpuscle, Pacinian corpuscle, and the Ruffini endings. So, where they are presentation is also asked in the examination. So, Merkel disc is present in the superficial. This is present in the superficial, but they act very slow. They act very slow. Messner corpuscle also is present in the superficial. They act very fast. Okay, Pacinian corpuscle present in the D, they act very fast. Ruffini endings is present in the D, they act very slow. What about the examples? What they are examples of? So Ruffini endings is for the stretch, Pacinian corpuscle for the high vibration and pressure, high pressure and vibration, and superficial or the fast. This Messner corpuscle for the light touch. This is for the light touch, and Merkel disc is for the edges, 2 points, 2 points discrimination, and also Braille. You might have heard the Braille lippy that is used for the blind people. That is for the Braille. Okay, these are the skin important receptors that is present in the body. Right. Next, this is the tongue. Tongue receptors is also commonly asked. This is the bitter. This is the umami, and here you are going to see this is the sour. This is the sweet, and here it is the salt. So what is the receptors for the salt? Salt itself is a sodium, right? So we have the sodium channel, and for the sour we have the bicarbonate channel, and also some of the very little sodium channels. And for the sweet we have the TRP channels, T1, T1, R2, R3. Just remember like this. And for the bitter we have the T2R receptors, and for the umami, that is monosodium glutamate. Monosodium glutamate is present for the umami. Okay, that is for the umami. This is the important points that you have to take note. Next, coming to the action potential. So whenever you see action potential, there is a important phases such as 5 phases that we are discussing. Image based question can be also asked here. So what are those 5 phases? So in the first phase, here you can see this is the first phase, right? So here in the first phase, there is an entry of the sodium. Okay, the slow entry of the sodium. Once the its entry is came, once they have the threshold is reached, suddenly there will be increased sodium. Okay, this is a fast influx of the sodium in the second phase. Why? It has to create the depolarization. Next, after it reaching the point of the 40 threshold, what happens? It is going to, the potassium is going to out. Okay, this is for the repolarization and back to the stage. And if still the potassium is slowly is going to release, it going to cause the hyperpolarization. And at the resting stage, again there will be inside the 2 potassium, and there will be out of sodium, right? This will back to the resting state. This is what the total action potential. Just revise with me, what are the phases, guys? The first phase, there will be slow sodium influx. Second phase, there will be increased sodium, like fast sodium influx. So this is where the depolarization phase will be seen. And the third phase, here the potassium is out. Here you are going to see the repolarization. And there is a fourth phase, that is potassium is out but slow. This will cause the hyperpolarization, more negative in the cell, and there will be back to the resting state. These are the important phase of the action potential of the nerve. and they are some of the laws that is present. One is the all or none. One is the all or none law. What is mean by the all or none law? If there will be the action potential or there is no action potential at all. If there is action potential, all will be having the action potential. If one is not have, there is nothing will be having. That is what the all or none law. Next, absolute refractive period. In the absolute refractive period, even if you give the stimulus also, even if there is a stimulus also, there will be no change. And refractive period, the other one is the refractive period, and in the refractive period, sorry, this is the relative refractive period. If you give the stimulus, okay, there will be the action potential. Again, the presentation will be seen. So one third of the cycle will be contents of the absolute refractive period, whereas this part will be contents of the relative refractive period. Okay, this is what the thing that you have to remember. And some of the classifications, what are the inhibitory type, what are the inhibitory type? So the inhibitory type are the GABA. Most importantly, how the GABA will be working, guys? So there will be influx of the chloride and efflux of the potassium, potassium out and chloride in. So this is how the inhibitory function will be seen. Excitatory, how? So excitatory will be with the help of the glutamate, such as the glutamate. So what happens here? Potassium out is slow, whereas sodium or calcium in. This is what happens in the excitatory of the inhibitory. And I want to guys to know, understand, if there is an increased calcium, the cell will be excited. If there is a decreased potassium, the cell will be non-excited. Okay, this also has to be take note, has been asked in the examination. Right? Next, we are going to discuss about the Erlanger-Gassett classification for the nerve fibers. This is for the both. One is for the sensory and other one is for the motor. Okay, so alpha, A, B, C, they are the types. Alpha type is for the muscle spindles. Okay, this is for the muscle spindles, and also alpha motor neuron for the skeletal muscle. Okay, and it will contain contents of the maximum fibers and maximum nerve conduction will be seen. Alpha beta, this is for the touch and pressure, and alpha gamma is for the muscle spindles, but it is for the motor type. And alpha delta, this is for the temperature and the pain, and beta is for the sympathetic preganglionic, and for C for is the sympathetic postganglionic and the cutaneous pain. So here this is the slow pain, and here it is the fast pain. Okay, this point has to also be take noted. Next, we have to discuss about the pain receptors present that is present in our body. One is the fast pain which will be contents of the, here you can see this is one, A delta. And slow pain, slow pain is caused by the C fibers. Why it is slow? Because it is a non-myelinated. And what is the spinothalamic tract for this? Here it is the neospinothalamic tract. Neospinothalamic tract. And here it is the paleospinothalamic tract. This is for the spinothalamic tract. So can we decrease the pain? Yes. How? By massage. And massage, what is going to do done by the massage? So massage is actually going to release the A beta. These A beta which will be going into the spinal cord, and from the spinal cord it will inhibit the main A delta and C fibers. So what is the important present in the massage or the touch? That is the A beta. This question has been also asked in the examination, so please don't ignore this. Okay, next moving on to the tracts that we are going to discuss. So first we are going to discuss about the spinothalamic tract. Okay, spinothalamic tract. So in the spinothalamic tract, I want you guys to take note that this is the spinal cord, this is the thalamus, and this is the cerebrum, right? So the decussation where it is happening from the spinothalamic, So name itself telling it is spinothalamic, it is descending upward, right? So from the spinal cord it is going to decussate here in the other side, and from there it is going to upward. Sorry. So from the spinal thalamic tract it is going to upward. This is what happens in the spinothalamic tract. So where is the decussation is happened? Here. The site of the decussation is happened at the spinal cord itself. So what are the functions of the spinothalamic tract? This is for the crude touch, pressure, and also pain, temperature. Next, dorsal column tract. Dorsal column tract. So again CNS, medulla, and then we have the spinal cord. How it is going to decussate? So it seems the dorsal column tract also coming from the spinal cord, and but it is going to decussate at the medulla. So the decussation is took part in the medulla. Okay, so here it is. So the decussation is placed at the medulla. That is what in the dorsal column tract. Now what are the functions of the dorsal column tract? Stereognosis, and 2 point discrimination, and also touch, light touch, that will be seen here. Light touch is also seen here, and proprioception, and vibration. This is seen in the dorsal column tract. Next, we also have the other tract, that is the corticospinal tract. Corticospinal tract, which will be contents of the name itself telling cortex to the spinal. So this is a type of the motor pathway. Okay, so from the brain to the medulla and the spinal cord. So this is going to, again the decussation is going to be here only, in the medulla only, but it is coming as an ascending tract here only. I'm sorry. So the decussation is took place at the medulla only. Okay, that is a corticospinal tract. Most of the motor actions will be coming from the corticospinal tract. So what happens, why these tracts are very important? Because we have one syndrome. That syndrome is known as a Brown-Sequard syndrome. So if you cut Brown-Sequard syndrome, if you completely cut the one part of the spinal cord, what happens? So if you cut the one part of the spinal cord, here you can see spinothalamic tract. Here the decussation itself completely on the other side, right? The right side tracts are going on to the left side tracts are on going on to the right side. So what happens here in the Brown-Sequard syndrome? The spinothalamic tracts, there will be contralateral changes. Contralateral changes will be seen at the spinal tract. Whatever the corticospinal tracts and whatever the dorsal column tracts, so the dorsal column tract and also corticospinal tracts, so these will be having the ipsilateral changes. So there will be changes of the ipsilateral changes that you are going to see. Okay, that is what the most important, that you have to take note about the Brown-Sequard syndrome if the half of the part will be cut out. Next, nuclei. Hypothalamus nuclei and their function. Anterior is for the heat, posterior is for the cold, lateral is for the feeding, and ventromedial is for the satiety, suprachiasmatic circadian rhythm, supraoptic ADH or the vasopressin, paraventricular for the oxytocin, ventrolateral preoptic is for the sleep, mammillary bodies is for the memory. And the Brodmann areas, visual motor is the 7, the visual cortex is the 17, primary motor is the 3, and primary gustatory is the 43, primary auditory is the 41 and the 42. And whatever the primary sensory, that is 1, 2, 3, and this is the primary sensory. And we also have the Brodmann's, sorry, Broca. What is the Broca's number? 44. What is the Wernicke's? Is the 22. This is also important Brodmann's areas that you have to remember. Next, some of the important neurotransmitters and our basal ganglia structures. Substantia nigra. Substantia nigra, which will be contents of the GABA, and the disease or the lesion that will be, sorry, substantia nigra is not the GABA, guys. Substantia nigra is actually dopamine. So if there is a decreased dopamine, the patient will be having the Parkinson's, right? Next, we have the caudate. Caudate is having the GABA. If the GABA is decreased, the patient will be having the chorea. Next is subthalamic nucleus. Subthalamic nucleus. This is contents of the glutamate, and if there is a lesion that is present here, that is the hemibellismus. Hemibellismus. And next we have the globus pallidus. Globus pallidus. This contains of again GABA, and if the GABA is decreased, that is the athetosis. That is the athetosis that will be present here, right? So these are the important points that you have to take note, right? These are the some of the important points of the central nervous system, the lesions, the neurotransmitters, and their actions and the classification. Okay, next, moving on to the muscle physiology. Okay, in the muscle physiology, so the first one that we are going to discuss is the muscle contraction, right? The muscle contraction and the structure. So these are the two Z lines, and this is the thick filaments, which is also known as a band, and this is the thin filaments, these are known as a I band, and this one is the middle one, this is the M point or the H zone. What is the importance of the H zone, guys? The H zone is the non-overlapping zone. Okay, this is the sarcomere. What is the importance of the sarcomere? This is also known as a functional unit and also structural unit of the muscle. That is the sarcomere. This is play the major role in the most of our body function, right? Next, we also have the nebulae. This is what the nebulae. Okay, this is the nebulae, and here the myosinocin that is going to present. Next, we also have the titin which will be connecting to the sarcoplasma, and there will be the structural and some of the contractile also. Okay, let's see here. What are the contractile proteins? Contractile will be the myosin and the actin. What are the supportive? That is titin and dystrophin. So if there is a deficiency of the dystrophin, you are presented as a DMD, Duchenne muscular dystrophy. If there is a decreased titin, you will be presented as a dilated cardiomyopathy. These both are very important questions that has been asked. So what happens first? Whenever you see, so during the muscle contraction, what happens? What are the changes that you are going to see? Before that, we have to understand about the what is the neuromuscular junction, how the action potential is generated, and how the muscle contraction is seen. So there is a influx of the sodium channels. So there is an influx of the sodium. After that, the threshold of the inside will be increased. So this will cause the sodium influx. Voltage gated calcium channels will be inside efflux. Inside it is going to come on. After coming into the inside, this is the presynaptic. After this, calcium will form the vesicles. These vesicles came out with the help of the exocytosis of the calcium channels. So these contains the acetylcholine. This acetylcholine is going to touch the receptors. These are the nicotine receptors or the muscarine receptors. This will cause the influx of the sodium. This will cause the influx of the sodium. Okay, so this is the voltage gated, here also voltage gated, but this is the ligand because this is touched the receptor. So these are the ligand gated sodium channels. After coming into the sodium channels, what happens? Here the action potential is generated. Action potential is generated and it will enter the T tubules. T tubules it is going to enter. These T tubules which will be contents of the this structure on the muscle. This structure is known as a dihydropyrimidine. Dihydropyrimidine. And these dihydropyrimidine which will be contents of the another structure, that is the ryanodine receptor. That is a ryanodine receptor. So once this action potential is entered, so these dihydropyridine which will be helping the ryanodine receptor. So this is like a lock and this is the key. Okay, this theory is known as a lock and key theory. What happening in the lock and key theory? So once the action potential is touching the dihydropyridine, it is going to touch the ryanodine receptor, and these ryanodine receptor now release the calcium. This ryanodine receptor will release the calcium, and that calcium is going to cause the muscle contraction. That calcium is going to cause the muscle contraction. How the muscle contraction is going to take place? So first, basically, there is a first thing that is going to happen is the ATP touching. Okay, so the ATP is going to bind. ATP is going to bind with the myosin head. Once they are touched with the myosin head, it is going to cause the hydrolysis of the ATP. It is going to cause the hydrolysis of the ATP. ATP is converted back as a ADP. After that, it is going to form the cross bridge formation. Here in the cross bridge formation, it is going to release the calcium. It is going to release the calcium, and this is known as a muscle contraction. This is known as a cross bridge formation, where it is going to connect with the calcium of type of the actin, and this is going to form the cross bridge between the myosin and the actin. And once there is a power stroke, the ADP will be go off. It is going to be slighted off, and again it is going to wait for the another ATP to form and another ATP to attach to the myosin head, and the cycle will be continuous here. Why the rigor mortis will be seen? Why the rigor mortis is seen? Because the ATP is depleted, right? It has to be touched with the ATP. So in the rigor mortis patients, there is no ATP. So that's why it is going to be seen with the ATP depleted state. The patient will be having the stiffening of the muscles. Okay. Next, actin will be contains of the three filaments. One is the troponin I. Another one is the tropomyosin, and next one is the troponin C. So troponin C, which will be contents of the calcium. Troponin I is for the MI marker we are going to use. Right, these are the important parts of the actin. Next, what are the changes that we are going to see in the here in this particular structure? So here we are going to see whenever there is a action potential, the A band is going to be intact. Here you can see this A band is going to intact. Okay, next, Z lines, these two Z lines which will come together, Z lines will come together, I will be going to shortens, H or the M, they will disappear. These are the changes that will be presenting in this particular structure. Okay, next, moving on to the here. And another point that I wanted to tell you guys, here you can see this ryanodine receptor. What is the importance of the ryanodine receptors? If there is a ryanodine receptor is not present, so there will be no muscle contraction, the patient will be having the death only, right? So if there is a ryanodine receptor is present but aggravated, so this will cause the one condition that is known as a malignant hyperthermia. That is malignant hyperthermia. So these malignant hyperthermia is due to excessive of the ryanodine receptor. So that means more and more muscle contraction, which will increase the BP, which will increase the heart rate, increase the temperature, and also muscle breakdown, which will be going to cause as a rhabdomyolysis. So this is going to cause the renal failure. So what is the drug of the choice for this patient? Dantrolene. Immediate dantrolene is given for this patient as a treatment of the choice. Next important graphs that has been asked in the examination. Let's say this is a point A and this is a point B. Point B, this is the point C, and point D. This is between the force and the length. We are actually checking for the tension. If there is a severe overlay, For example, point A. Point A have no tension. Why no tension? Here you can see the overlap will be more, right? Overlap is too much. That is why there is no tension at all. Next, point B and the point C. The overlay is very less. So here we are going to see the overlay means that is between the two sides of the contraction I band and the A band. So here that will be presented as a maximum tension. Okay, same with the C also, it will be going to have the maximum tension with the overlay of the I band and the A band. Next, point D is having the minimum tension. It is going to have the minimum tension because the overlap is very long. That is why it is going to have the minimum overlap. This is one of the graph that is already asked in the examination. Okay, next, coming to the chronaxy and the rheobase. What is meant by the rheobase, guys? Rheobase is the minimal stimulus. I gave the minimal stimulus, that is known as a rheobase. And what about this one? This is the utilization time. So when I gave in the normal stimulus, what is the time that has been taken? That is known as a utilization time. Next, two times of the rheobase, double the stimulus that I gave. So when it is then generated, that is the chronaxy. Chronaxy is nothing but two times of the stimulus response is the chronaxy. Two times of the stimulus response is the chronaxy. This graph is also asked previously in the examination. Next, let's know about some of the important parts of the um, nerve potentials. For example, what is the neuron potential? Is minus 70. And how about the cardiac muscle? Cardiac muscle or the SA node? This is around minus 90. Same as the skeletal muscle also, which will be contains of the minus 90 millivolts. And potassium is 90. Smooth muscle, which will be contains of the minus 50. RBC is minus 12. Okay, sodium is plus 60. Calcium is plus 130. Okay, these are the some of the important, and chloride is minus 70. These are the nerve potentials and the equilibrium membranes. Next, three types of the muscles and their main function that I want you guys to understand. So what is this muscle, guys? There is a sarcomere and there is a multiple striations that has been seen here. This is a skeletal muscle. And here you are going to see intercalated disc and the gap junction. This is the cardiac type. And here it is a smooth muscle. So what are the importance of this? This is a striated. Here also it is a striated. This is the non-striated, right? This is the voluntary. This is the non-voluntary. Here also it is a non-voluntary, right? Main functions of the skeletal muscle. There is a abundance of the ATP that is present here. Abundance of the ATP presentation will be seen, and it is mostly calcium dependent. Okay, and here also ATP presentation is seen with the calcium plus calcium only. The combination will be here the calcium with colmodulin. This will be presented with the colmodulin, and here we are going to have the actin and myosin, right? Calcium and calcium, that is the cardiac muscle. And next, in the skeletal muscle, we are going to see the muscle contraction with the help of the multiple cross bridge formation and also the multiple ATP formation that is going to see here. And it is contents of the many structural. For example, titin, it is contains, and also sarcoplasmic reticulum, it is presented. And next, we also have the dystrophin, right? Here also the titin will be presented, and here no titin and no dystrophin. What then? What are they going to have? Instead of that, they have the dense bodies. Dense bodies they are going to present. Here it is a cross bridge formation. Here it is a cross bridge formation. Here it is going to cause a latch bridge formation. Latch bridge formation you are going to see here in the smooth muscle, right? These are the important points that you have to take note. In the smooth muscle, another point, there is a interstitial cell of Kajal That is the main pacemaker of the cardiac, sorry, in the smooth muscle. Where is the tetany is presented? Here tetany is presented, here tetany is absent. Tetany is absent in this particular cell. Alright, next moving on to the different types that I want you guys to understand here of the stretch reflex and the Golgi tendon reflex. So the difference between the stretch reflex and the Golgi tendon reflex. Stretch reflex, whenever you do the muscle lengthening, you are doing the muscle length, this will activate the muscle spindle, and once the muscle spindle is activated, it is going to activate the 1A and the 2 muscle fibers. This is the afferent, and now it will go into the spinal cord. Now the spinal cord will activate the alpha motor neuron, and these alpha motor neuron will cause the contraction. So the muscle spindle here is actually triggered by the muscle lengthening. What about the Golgi tendon? Golgi tendon is about the muscle tension. It is about the muscle tension. Once it is increased, it will activate the Golgi tendon, and these Golgi tendon will increase the 1B. This will increase the 1B. This is the efferent, and from here it will be reaching the spinal cord, and spinal cord will again activate the alpha motor neuron. This will cause the relaxation. And here the alpha motor neuron, this alpha motor neuron is the efferent. This is the efferent. So this is difference between the stretch reflex and the inverse stretch reflex. Okay, this is the monosyntonic, this is the monosynaptic, this is a disynaptic. Remember 10 10, tension and lengthening is the muscle spindle. Okay, next moving on to the next topic, that is a respiratory system. So in the respiratory system, first we will understand what is the inspiration, expiration and the forced expiration. So first we have to see about the inspiration. Whenever you are inspiring, this is an active process. Okay, during this you are going to use the main muscle, that is the diaphragm. And along with the diaphragm, what happens during the inspiration, the volume is more but the pressure is less, right? Next, during the expiration, what is this is a passive process, and you are using the intercostal muscle. Here the volume is less but the pressure is more, right? Next, forced expiration. What happens in the forced expiration? This is also active. Now you are using the abdomen muscles. Active process means the volume is more and the pressure is also more. That is what the forced expiration, and most of the times the volume will be less in the forced expiration. This is the main mechanism behind the all three, the inspiration, expiration and the forced expiration. Next, see the spirometry. In the spirometry, this is the tidal value, right? This is the tidal value, and tidal value is the normal inspiration and the expiration, which is around the 500 ml, and maximum you take the inspiration. This is the inspiratory reserve value around the 2.5 liters. The maximum that you going to expire, this is the expiratory reserve value around the 13 1.3 liters. Okay, 1.3 liters that will be presented, and whatever that left after all the air that you are expired, that is the residual value. This is also around the 1.3 liters. All this if you combine, you are going to get around the 6000 liters or the 5.5 liters that you are going to get. And whatever the capacities, inspiratory capacity, which will be including of the inspiratory reserve value plus the tidal value. Okay, next, vital capacity includes the IRV plus RV, plus the TV plus tidal value plus expiratory reserve value. Total lung capacity, which will be including the vital capacity plus the residual value. Next, forced residual value. It is going to include the ERV plus the residual value. Okay, this is the important points. Next, forced expiration at 1 second. Forced expiration at 1 second, it is going to only take out the 74%. Only 74% is exhaled out. FEV2, it is going to be the 94% exhaled. At third second, it is going to be the 97% exhaled. This is what the mostly that we are going to see in the capacities. But what is the test that we are going to do here? The most important test is the spirometry. But there is a spirometry have the one of the problem that it cannot calculate the RV residual volume, forced residual capacity, total lung capacity. For them, we can use the body plethysmography. For them, we are going to use the body plethysmography. Body plethysmography we have, and also helium dilution method. These both are used for the RV and the FRC and the TLC. Next, moving on to the pressure versus the value. Okay, so whenever you are expiring or exhaling outside, so the curve will be on the upper side, means the compliance is more. Okay, so the compliance is more, but during the inhalation the compliance is less. Why this compliance is difference between this? So the compliance is equal to the volume by pressure. So for example, whenever you are inspiring the air, the volume is more. So what happens? The compliance is decreased. Why? Because the surface tension is very far away from each other. The compliance is decreased and the surface tension is more. Right? Here the surface tension is more. The surfactant is less because of volume is increased. Whereas expiration, when you are expiring the air, the volume itself very less. So the surfactant is very near to each other. The volume is decreased, the surface tension is decreased, so that the compliance is also increased. Okay, the compliance also here in the inspiration compliance decreased, and here the expiration compliance is increased. That is what this graph is going to explain us. Okay, next, compliances. Again the compliance about only, there will be always collision between the expanding forces and the collapsing forces, and everything will be ending with the forced residual capacity. And how? The first the chest wall, which will be having this curve, that they raise a volume of the air in the lungs after the expiration, the collapsing force after the forced residual capacity. And here this is a both, and this is the lung and the chest wall. This is the lung and the chest wall. This curve, this is the lung and chest wall, and this is only the lung. Okay, so this is the chest wall, here you can see, and this one is the both, and here this one is the lung. These are the compliances that has been asked in the last year examination only. Okay, that you should not be missed this. Please do read that one. Next, oxygen capacity. Right, this question is also very much important. So before the oxygen capacity, one hemoglobin is going to take the 1.34 ml O2. It will be going to carry. That means 15 hemoglobin is going to carry the almost 20 ml of the oxygen. Right, this is what. Next, hemoglobin plus O2, which is known as a oxyhemoglobin. Hemoglobin plus carbon monoxide, which is known as a carboxyhemoglobin. Hemoglobin plus CO2, which is known as a carbamino. Hemoglobin plus ferric, that is a methemoglobinemia. Right, next. So at this point, there will be 25 mmHg, and at the P50 level, this curve is known as a sigmoid curve. So whichever the right side shifting, that is decreased affinity to the O2. So they are going out of the O2. Okay, they are going away from the O2. So here the P50 will be increased. Whichever going on to the left side, they are very highly attached to the O2. So that means it is going to be decreased P50 levels. Okay, this is the decreased P50 levels. What is the examples? There is an increased acidosis in the body. There is an increased hyperventilation, increased 2,3 dpg, or increased hypercarbia or the temperature. All will be seen in the right side of the shift, and left side of the shift, which will be more affinity. So that means fetal hemoglobin, temperature 2,3 dpg is less because of the blood products that we are giving to this patient, and alkalosis and PCO2. The carbon dioxide is completely decreased. This is what's seen in the left side of the shift. Next, ventilation, perfusion, pressure. So generally, the ventilation that we are going to calculate is the 350 ml of the body only. So 350 ml only we are going to calculate. Why? Because the 150 ml is a dead space. So the 150 ml is a dead space, and this is only the one which will be exchanging. Okay, so 350 ml into number of respiratory rate, so around 12 we are going to get. So this is the 4.12 ml. This is the ventilation, and perfusion is around we are going to have the perfusion will be mostly like 5 liters, let's say. So ventilation by perfusion, so ventilation by perfusion, we are going to get the 0.8. This is considered as a normal. This is considered as a normal. So when there is a, this is the normal one, we are going to see if there is a high VQ match or it is an infinity, that is the problem is in the perfusion. That is problem is in the perfusion, pulmonary embolism. Next, the low VQ, that means zero VQ. The obstruction is in the ventilation, like airway obstruction and the pneumonia. This is the examples of the ventilation and the perfusion. Next, this is the normal respiration. You are going to see the normopnea, expiration, inspiration, expiration, inspiration. Next, Boyd's breathing. What is the Boyd's breathing? There will be the hyperopnea, completely hyperopnea and pause, apnea. Hyperopnea, pause. This is seen in the neuron damage, especially in the medulla, and this is also associated with the meningitis. Next, Cosmol's breathing, complete the hyperopnea. This is seen in the sepsis, aldehydes, lactic acidosis, DKA, and diabetes ketoacidosis patients. Chinese Stokes. Chinese Stokes will be the hyperopnea, hyperopnea, apnea. Okay, sorry, hyperopnea, hypopnea, apnea. Hyperopnea, hypopnea, apnea. So this is seen in the sleep. This is the seen in the like sleep patients, hypoxia patients, and also hypoperfusion to the brain. This is known as a Chinese Stokes. This question is also has been asked in the examination. Next, we have the pons. So in the pons, we have the two centers, right? Next, we have the medulla. Medulla also we have the structures, right? So first let's see one by one. What are these structures? So in the pons, this is known as a pneumotoxic, pneumotoxic, and this one is the apneustic. This is the pre-Botzinger. This is the DRG, and this is the VRG. This is the VRG. So what are these? Let's see. So this pneumotoxic center, which will be helping for the inspiration. Okay, it will be inspiration. It will decrease the limitation of the inspiration. Apneustic will increase the depth of the inspiration, and VRG, it will be important for the expiration. It will be important for the expiration, and DRG, again this is also important for the inspiration. Whatever the pre-Botzinger. Pre-Botzinger is an pacemaker of our body. And how it is a pacemaker? Because it will be giving rise to the, whenever you sleep, you can't keep the your consciousness to the respiration, right? So this will maintain that respiration. smooth maintenance which will be done by this pacemaker. That is a pre-Botzinger type. That is a pre-Botzinger type. Next we have to discuss about the another important thing. Let's discuss here. So for example, this is the Nemo site. Okay. So this is the type 1, this is the type 2. What is this type 1, type 2? Type 1 pneumocyte, if you can observe, they are long and they are squamous, important for the gaseous exchange. And here, this is the type 2 pneumocyte, this is the cuboidal, and less, these are many, and this is important for the surfactant. So what is this surfactant? Let's discuss here. What is the surfactant? Surfactant is important for the respiration. How? It will increase the compliance of the respiration by decreasing the surface tension. By decreasing the surface tension in the body. It is going to present by the 35 weeks of the age of gestation. And how we are going to check, guys? Because it contains the mostly will be the dipalmityl phosphatidylcholine and sphingomyelin. So when we check this ratio in the baby, if it is more than 2, that is normal. If it is less than 2, that will be the abnormal, presented as a respiratory distress syndrome. And what is the defect that is presented by the elderly patient? Upper respiratory or the acute respiratory distress syndrome that will be present in the children. Sorry, in the elderly people. Next, let's see the hypoxia types. In the hypoxia types, we have the 4 types of the hypoxia. Hypoxic hypoxia, there is a decreased O2. Anemic hypoxia, there is a decreased RBC or decreased hemoglobin that we are going to see. And stagnant hypoxia, the flow is abnormal here. And the histotoxic hypoxia, the tissue itself is damaged in this patient, right? And what is the treatment for this patient? Just give the O2. And here the treatment for this patient, treat the underlying cause, mostly anemia. And here also the treatment will be the treat underlying cause, and here also the treatment will be treat the underlying cause. Okay, next, DLCO. What is this DLCO? Diffusion of the carbon monoxide. The carbon monoxide itself the 2 times of the high flow to the RBC, right? So the DLCO will be high in the polycythemia patients and also in the exercise patient, and it will be decreased in the anemia patients and also decreased in the aging patients. This is the one that you have to remember about the DLCO also. Okay, next, mostly these are the topics that will be asked in the examination, guys, that you have to take note. Next, moving on to the cardiovascular system. In the cardiovascular system, first you have to take note about the action potential of the pacemaker. This is the SA node. Okay, here the T type of the calcium channel is going to increase. This is the 0 phase. Sorry, this is the 3rd phase. This is the 4th phase. This is the 0. This is the 3rd phase. So the calcium channels is going to influx, and once they are influx, there is a depolarization. Okay, and next the potassium channels are going to outside. That will be the repolarization phase. Only there are 3 phases in the pacemaker of the SNO. And in the ventricle, let's see in the ventricles, there are the 4 phases. This is the 4th phase. This is 0, 1st, 2nd, 3rd, and the 4th. Here there will be influx of the sodium, and here also sodium influx. We are going to see the depolarization. There is a potassium out. This is the early repolarization, and still potassium is going out, but in the 2nd phase there is a calcium influx. So this is known as a plateau phase. And here the again sodium is going, sorry, here the potassium is came out. Potassium is out. There is a repolarization that we are going to see. All right, next, again come back here. 0 phase is for the T type of the calcium channel in. Third phase is for the potassium out, and the fourth phase is for the sodium channel in and also the calcium channel inside. Next, zero phase is potassium in, the first phase potassium out. This is the early. Second phase potassium out and calcium in. Third phase is mostly potassium out, and the fourth phase potassium in and the sodium out. So to maintain the resting membrane potential. Next, how this complete pathway is seen from the SA node to it is going to be seen in the AV node, and from the AV node it is going into the bundle of His, here the bundle of His, and then it is going into the Purkinje fibers, and Purkinje fibers will be going to the left bundle branches and the right bundle branches that we are going to see. This is how the conduction pathway will be seen. This is known as a dromotrophy. What is a dromotrophy? Conduction. What is the inotropy? Inotropy is the contraction. Let's see the conduction and how is the electrocardiogram is formed. So P wave is for the atrial depolarization. QRS complex, this is for the ventricular depolarization, and for the T wave is for the ventricular repolarization. Next, we have the segments. What are the segments, guys? Segments is nothing but there is no action is happening. For example, PR segment, ST segment. So this is for the AV delay, and this is for the J point. Next, we also have the intervals. What is the intervals is going to be RR interval, that means 1 beat, and QT interval, which will be ventricular depolarization, ventricular repolarization, PR interval, which will be atrial depolarization and atrial repolarization, and also AV delay, that is seen in the intervals. And what are the leads that we are going to see? Chest leads is going to be 1 to 6, and augmented leads, which is also known as a unipolar leads, that will be your AVF, AVL and AVR, whereas bipolar leads 1, 2 and 3. This is the Einthoven triangles that we are going to see. Next, coming to the JVP, how we are going to see a JVP? A wave, atrial contraction, C wave, that is for the ventricular systole, and here you are going to see the tricuspid bulge, and the X threshold, this is for the ventricular systole, and maximum filling that we are going to see is the V, this is for the atrial filling, and emptying of the atrium, that is seen by the Y form that we are going to see. And what are the pathologies? If you have to see the pathologies, for example, absent A wave is absent, Canon A waves, and Y descent is absent, more prominent V, more prominent X, and giant A wave. Absent A wave is seen in the AFib. Canon A wave is seen in the complete heart block. Y descent is absent in the cardiac tamponade. Prominent V wave is seen in the tricuspid regurgitation. Prominent X wave is seen in the constrictive pericarditis, and the giant A wave is seen in the tricuspid stenosis. These are the pathologies that you have to remember. Next, moving on to the cardiac cycle. How the cardiac cycle is presented is, cardiac cycle is generally 0.8 seconds only. Cardiac cycle is generally 0.8 seconds. So first, we have the isovolumetric contraction. Once the isovolumetric contraction is seen, there will be rapid ejection and there will be slow ejection. Okay, there is a slow ejection. This is known as a systole phase. Systole phase. During the isovolumetric contraction, what happening? Isovolumetric contraction, the ventricles pressure is opening, and here the S1 sound is came. Why the S1 sound is came? There will be closing of the mitral and tricuspid valve is closed. Here the S1 is formed. Rapid ejection, here the atrial and the pulmonic is open. Next, isovolumetric relaxation. Isovolumetric relaxation. This is the isovolumetric relaxation. What happens here? There is a S2 formation, means atrial and the pulmonic will be closed, so that there will be filling, rapid filling and slow filling, and there will be atrial contraction. This is known as a diastole, and this is the active. This is known as a diastasis. Okay, this is the cardiac cycle, guys. Okay, first systole, second continued by the diastole. And what is the end systolic volume? End systolic volume, which will be 50 ml. End diastolic volume, which will be 120 ml. End systolic volume. Stroke volume, how we are going to calculate? End systolic volume by the end diastolic volume. Ejection fraction, how we are going to calculate? Systolic volume by the end diastolic volume into 100. Cardiac output, you are going to calculate heart rate into the stroke value. These are the points that you have to take note about the different of the equations of the cardiac cycle. All right, next, how about the murmurs? So just remember isovolumetric contraction, isovolumetric relaxation, isovolumetric contraction. Okay, so this means this is the systole, this is the diastole. So whenever in the systole, all the systoles will be atrial stenosis, pulmonic stenosis and mitral regurgitation, tricuspid regurgitation. All these are the systolic murmurs. Next, opposite to that, atrial regurgitation, pulmonic regurgitation and mitral stenosis, tricuspid stenosis, these are the diastolic murmurs that you have to remember as an easy mnemonic. Next, baroreceptors. What are the baroreceptors? They present in the aortic arch and the carotid sinus. In the aortic arch, this will be run by the vagus nerve. In the carotid sinus, this will be run by the glossopharyngeal nerve. So whenever there is an increased BP, what happens? These baroreceptors will be activated. These baroreceptors will be activated, and baroreceptors will be sensing from this one, and they are going to send the signals for the 9th nerve and the 10th nerve. These 9th and 10th nerve will act on the two waves. This will be acting on the two sides, and one is will be the rostroventrolateral medulla, where it will be decreasing the sympathetic system, so that the BP will be decreased, and it will be activating the parasympathetic nervous system. How? With the help of the nucleus ambiguous, so that the heart rate is also decreased here and also BP will be decreased. So this is the main mechanism between the baroreceptors that you have to remember. Next, pressure and the volume graph. How we are going to remember? This is the pressure, this is the volume. If the pressure is dropping, this is the isovolumetric relaxation. If the pressure is increasing, this is the isovolumetric contraction. Where is the S1 is seen, guys? Where is the S1 is seen here? This is the S1, and here this is the S2, right? So this means the volume is improving. So this is the rapid filling and the slow filling, right? This is the ejection is seen. So this is the rapid ejection, this is the slow ejection. These points that you have to take note between the pressure and the volume graph. Next, RAS system. Whenever there is a renal perfusion is decreased, whenever there is a BP is decreased, renin is activated. It will convert the angiotensinogen to angiotensin 1, and the angiotensin will be converted as a angiotensin converting enzyme to the angiotensin 2. and here it will be activating the aldosterone. Aldosterone will increase the sodium, aldosterone will take the more and more water. And what happens here? There will be increased thrust, increased vasoconstriction, and increased sodium absorption. So overall, it will increase the BP, it will increase the renal blood flow, and also it will maintain that complete the body by these mechanisms. Okay, these are the points that we have to note. Next, some of the reflexes that you have to remind. One is the Bain-Bridge reflex. Bain-Bridge reflex. So these Bain-Bridge reflex which will be present in the right atrium, especially whenever you are having the congestive heart failure, this will increase the heart rate by stretching the right atrium fibers. Next, Zere's reflex. Okay, Basalt-Zeres reflex. Basalt-Zeres reflex, this will be present in the right ventricle. This will decrease the heart rate. This will be decreasing the right ventricle and decrease the heart rate by decreasing the sympathetic nervous system. These two types of the reflexes that has been asked in the examination before. Now we will be discussing in the renal system. In the renal system that you have to take note about the nephron, right? Nephron is a structural, also it is a functional unit of the kidney. Nephron will be consisting of the filtration unit. So what does this filtration barrier which will be containing? Most important is the basement membrane. These basement membrane which will be contains of the negative value, so it will be repelling the other substances. Next one is the podocytes, which is also known as a food processes, and next we will be also having the endothelia. This is the three important structures that will be maintaining the barrier for the, especially for the filtration barrier for the nephron, right? Next, moving on to the glomerular filtration. What happens in the glomerular filtration? Basically, I'm going to draw you here. This is your Bowman's capsule. Next, this is your arterioles, glomerulus. All right. Next, let's see. So first, this is the glomerular hydrostatic pressure. Okay, from the capillaries, you are getting into the capsule, right? Next, how much is this? This is the 60 mmHg from the capillaries to the capsule. Next one. Here, this is the colloid osmotic pressure. So from the periphery, you are going into the glomerular, right? This is around 30 mmHg. Next, so this is from the capsule. From the capsule, this will be around 10 mmHg. From the capsule, this is around 10 mmHg. So how much total? Let's see. So glomerular hydrostatic pressure minus Bowman's capsule colloid pressure plus colloid hydrostatic pressure. So 60 minus 30 plus 10. That means around plus or minus 20 to 30 mmHg will be coming. So this is the net filtration of the glomerular filtration, right? So what is the normal GFR? That is the 125 ml per minute. 125 ml per minute that we are going to get. Next, diuretics action. So in the proximal convoluted tubule here, you are going to have the acetylzolamide, that is the carbonic anhydrases. Next, here, this is the osmotic diuretics such as mannitol, and ascending thick loop of limb, that is for the loop diuretics such as furosemide, and distal convoluted tubule for the thiazides, and the collecting tubule is for the potassium sparing diuretics. Let's see once again the same example, because this is the example that has been asked. So proximal convoluted tubule for the carbonic anhydrates. Carbonic anhydrase. This classification we will be reading into the pharmacology. Okay, don't worry. This is the loop of Henle. This is for the loop diuretics. Sorry, this is for the osmotics. Only the water. Mannitol, and here thick ascending loop, this is for the loop diuretics, and this is for the distal convoluted tubule for the thiazides, and this is for the collecting duct for the potassium sparing drugs, right? Next, main mechanism. What are the main mechanisms? PCT, which will be having the 100% absorption, 100% absorption, highly ATP containing because of highly mitochondria is present here. Almost all, you name the glucose is 100%, the water will be the 80%, and also all the minerals which will be going here, and SGLT2 that is present here, PCT. Loop of Henle, this is for the descending, is for the water reabsorption, and for the ascending, this will be presenting of the pump, sodium potassium chloride pump. And distal convoluted tubule, this is for the sodium and chloride pump. Collecting tubule is for the ADH and mineralocorticoids and also H2O. This is for the collecting tubule. What is the action of the aldosterone, guys? Let's say this is the aldosterone. Aldosterone will be enters the sodium and the water. In exchange, it will send the potassium out. So this channel is known as a Romke channel, and this channel is known as a epithelial sodium channel. Okay, the exchange of the aldosterone main action is that it is increase the sodium and the efflux of the potassium. That means secretion of the potassium and reabsorption of the sodium. Right. Next, moving on to the cystometrogram. What is the cystometrogram? Before we know about the cystometrogram, you have to know about the bladder nerves. So what are the bladder nerves that is involved? Sympathetic nervous system. Sympathetic nervous system. It will be from the L1, L2 and L3. Parasympathetic nervous system. This is from the S2, S3 and S4. And from the brain, especially in the lobes, there will be paracentral lobule. Paracentral lobule will be present. How the urine will be coming? How the urine will be coming? There will be the micturition reflex. Micturition reflex. So basically, whenever there is a bladder is filled, once the bladder is filled, it is going to send the signal to the efferent signals. How the efferent signals? Which one is going to receive this efferent signals? That is came to the pelvic nerves. Once the pelvic nerves is received, they are going to send for the parasympathetic nervous system, which will be contracting the muscles, such as muscle contractions of the root tracer muscles will be contracted. Then this will relax the internal sphincter, internal urethric sphincter. There you are going to sense the feel. And once you sense, you have to relax the external urethric sphincter so that you can pee. Right? So here the catch. I just wanted to tell you guys, one has to be the contract, then only we have to relax the extra urethral reflex, right? So remember, don't confuse. This question has been already asked the same way they have given. So first we have to contract the internal urethric sphincter, then only we can relax the external urethric sphincter to pee or the micturition. Now we will see the cystometrogram. Cystometrogram is such it has reacted. So we are going to check for the urge of the urinary. So this is the first phase. This is the first time you will be feeling the urge. Here you are going to feel the urge. And here the pressure only increase in the urge. First phase. This is the 1B or the second phase. What we can say here, the maximum is reached. So the volume is increased, but the pressure is still constant. Pressure is constant. When the pressure is exceeded more, here where you are going to take place the micturition. So this law is known as a Laplace law. This is known as a Laplace law. So the first phase urge, second phase the pressure is constant, third phase is the micturition. Right? This question has been also asked in the examination. Next, another, the defects that you have to take note. In the PCT, if there is a defect, the condition will be the Fanconi syndrome. Okay, the Fanconi syndrome. In the distal convoluted tubule, this is the Gitelman syndrome, and in the collecting tubule, it will be the Liddle syndrome. And here in the loop of Henle, this is the Bartter syndrome. Bartter syndrome. Next, acid-base imbalance. Acid-base imbalance, pH is 7.4, HCO3 that will be 24, and PCO2 will be the 40. Remember, 4 is the common to remember. Don't have to be remember the accurate number, at least you have to know about the normal. Next, let's see. If the pH is low, that means it is acidosis. If the pH is high, this is the alkalosis. So pH is low, HCO2 is low, and the PCO2 is also low. pH is high, HCO2 is high, PCO2 is high. So the condition here is a metabolic. So this is a metabolic acidosis. Metabolic acidosis, primary defect is acidosis. Here this is a primary defect is alkalosis. Next, let's see another example. Here the pH is low, means acidosis, primary defect. HCO3 is high, PCO2 is also high. So this is a respiratory acidosis. So remember opposite, okay, opposite to the pH and the other metabolites, that is the respiratory acidosis. Fourth one, pH is high but the HCO3 is low and the PCO2 is also low. This is a respiratory alkalosis. So in the recent INICT examination, they asked about the examples also. So you have to take note about the examples. So regarding the compensations in the Fmge, that is not frequently asked, so we can skip that. Metabolic acidosis examples are lactic acidosis, DKA diabetic ketoacidosis, and commonly the diarrhea. Patients will be presenting. Next, metabolic alkalosis. Metabolic alkalosis, most common is the vomiting and also aldosterone. High aldosterone, which will be causing the metabolic alkalosis. Next, respiratory acidosis. This is seen in the opioid suppository, patients with the COPD and also pneumonia. Next, respiratory alkalosis. Examples are hyperventilation and decreased oxygen, hypoxia. These are some of the examples. Now you have to know about the hagma and the nagma also. What is the hagma? High anion gap. Examples are alcohol commonly and diabetes. Alcohol commonly that we are going to see, diabetes ketoacidosis, and also renal failure, and also commonly seen with the renal failure, aspirin poison. Next, normal anion gap metabolic acidosis, NAGMA. This is seen in the renal tubular acidosis. Also, patients those who are suffering from the diarrhea, and then there is a Addison's condition, decreased Addison's, all the condition which will be associated with the NAGMA. This is the anion gap that we have to take note. Next, thyroid metabolism. In the thyroid metabolism, the first phase, the iodide, this is the iodide which will be entering from the diet. Once they entered into the iodide with the sodium and iodide import. This is the sodium and iodide import. This is the facilitated diffusion, guys. Again, this is a question also can be asked, facilitated diffusion. And here it is going to converted as a iodine. Once this is converted as an iodine, with the help of the iodine, it is going to enter. This is the follicular cell. It will be entered into the colloid. Here you can see the colloid, it is entered. With the help of the which enzyme? Peroxidase enzyme, it is entered into and converted as a iodine. And here this iodine will form the together with the help of the thyroid globulin. When these both are combined, they are going to form the monoiodothyronine and diiodothyronine. So they are going to form the T3, T4, like that they are going to form. Here you can see all the main enzyme in the thyroid synthesis, that is the peroxidase. That is the peroxidase. So this first one, this iodine conversion is known as a iodination. And thyroglobulin plus the thyroid, this is known as a organification. This is known as a organification. All right, next. Then this is known as a coupling reaction. This is known as a coupling reaction and come back to the circulation. Okay, this is the form of the all the thyroid synthesis. Next, the thing that you have to remember, T3 is active inside the coli, but inactive and less half-life inside the circulation, but more potent. Next, T4. What is the T4? T4 will be the active, more half-life, and more number. Okay, that is the T3 and the T4. How it is formed actually? With the help of the TRH, TSH, and the T3 plus T4. This is the hypothalamus, this is the pituitary gland, and this is the thyroid gland. Next, this question has been also asked image based question. So about the gonadotropins, hypothalamus which will be releasing the gonadotropins in the anterior pituitary, FSH and the LH. FSH in the males act on the Sertoli cells which will be producing the inhibin, and Leydig cells which will be producing the testosterone. Testosterone is important for the spermatogenesis. Next, iron metabolism. So whenever you are taking the iron metabolism, this is in the Fe3. This Fe3 which will be entering into the circulation, here you can see this is the duodenum. So once it is entered into the duodenum, it is going to act on the one such receptor, that is the DMTE. DMTE. So it will entering into the DMTE. Once it is entering into the via DMTE, this will be converted as a apoferritin. Okay, so for this is for the storage. This is for the storage, apoferritin. So once this ferritin or the ferroprotein, so remaining whatever the iron that is left, that is going to be going into the circulation. So this circulation is known as a ferroprotein. So via ferroprotein on the other end, basolateral membrane, there will be the ferroprotein that will be sent into the circulation. And once they reach the circulation, that is going to be sent as to the bone marrows with the help of the ceruloplasmin. Where the ceruloplasmin is coming? This ceruloplasmin is coming from the liver. And transferrin, why this transferrin is important? This is sending to the bone marrow. And how we are going to stop? If we want to stop this one, we can stop with the another liver enzyme, that is the hepcidin. So that is the hepcidin. So hepcidin will tell the ferroprotein to stop, so that the entire system will be blocked for a while and the excess of the iron will be decreased in the body. So this is what the iron metabolism. Okay, this is the iron metabolism. Just I wanted to know about the, tell you another one also. So dopamine. So generally the dopamine will block the prolactin, and prolactin is going to block the GnRH. Okay, and TRH is going to increase the prolactin, vice versa. This question is also has to be important for your examination. Dopamine will stop the prolactin, prolactin can stop the gonadotropins, and the TRH can be produced by the prolactin, right? These are the important points that you have to take note, guys. So those who are new to this channel, please do subscribe, and if you want to access to this PDF, please text me in the Telegram group and join the Telegram group, and please like, so that the more students will be reached. And stay tuned, remaining subjects will be also discussed in the coming days. Until then, happy preparation, guys. See you in the next video.